ಯಾರು ಹೊಡೆದಿದ್ದು? The result is announced. You have seen, students? ಸ್ಟಾರ್ಟ್ ಮಾಡಿ ಸರ್. ಆನಂದ್ ಸರ್ ಶುರು ಮಾಡ್ಬೋದು ಸರ್. ವಾಯ್ಸ್ ಬರ್ತಾ ಇಲ್ಲ ಸರ್. ಆನಂದ್ ಸರ್. ಮೈಕ್ ಬರ್ತಾ ಇಲ್ಲ.
ಸರ್ is it ಈಗ ಬರ್ತಿದೆಯಾ ಸರ್?
ಎಸ್ ಸರ್ ಸರ್. ಹಿಂದೆ ಶುರು ಮಾಡಿ ಸರ್ ಸ್ಟಾರ್ಟಿಂಗ್ ಇಂದ.
ಓಕೆ, ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ಸರ್ ಈಗ?
ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆ.
ಓಕೆ. So this chapter will be studying about different types of cells, that is prokaryotic and eukaryotic cell. In eukaryotic cell, about the plant cell and the animal cell, then about the organelles and about all the organelles. So we'll go through a quick review of the chapter. A cell is the fundamental structural and functional unit of all living organisms. So Robert Hooke was the first person to observe cells, but he observed only dead cells, that is the cork cells, whereas Anton von Leeuwenhoek was the first to observe living cells. Then the invention of compound microscopes revealed the detailed structure of the cell, that is all the cell organelles. So Schildeen and Schwann were two scientists who formulated the cell theory. So Schildeen observed plant cells were composed of cells. So plants were composed of cells. Then Schwann was the first person to report about the plasma membrane and the cell wall. So usually in NEET, what happens, we study about the plasma membrane and the cell wall, but we usually forget the scientist who has discovered it. So that would be given in the starting of this chapter. So please do remember the plasma membrane and the cell wall were discovered by Schwann. He proposed a hypothesis that animals and plants are composed of cells and products of cells. Then Rudolf Virchow slightly modified the cell theory. Then he said that cells came from pre-existing cells, that is omnis cellula e cellula. So the two statements of cell theory: all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. Then all cells will contain cytoplasm. It is a semi-fluid matrix where the cellular activities and the chemical reactions occur. This keeps the cell in the living state. Then ribosomes. These are non-membrane bound organelles found in the cytoplasm, chloroplasts, mitochondria, and on rough endoplasmic reticulum. So the smallest cells here are the mycoplasmas. The largest cell is the egg of ostrich. The longest cell is the nerve cell, and the size of the bacteria is 3 to 5 μm. Human RBCs are 7 μm in diameter. Go to the next slide. So there are two major types of cells that can be classified as prokaryotic cells and eukaryotic cells. Prokaryotic cells, as the name says, pro means primitive, karyon means nucleus. They have a primitive nucleus without a nuclear membrane and a nucleolus. Neither do they have membrane-bound organelles. Membrane-bound organelles. They include bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. So, mycoplasmas are the smallest living organisms in the entire world. Then, after this, they are generally smaller and they multiply rapidly when compared to eukaryotic cells. So they vary in shape and size. So remember this, there are four basic shapes of bacteria: Bacillus, Coccus, Vibrio, and Spirillum. Bacillus, Coccus, Vibrio, and Spirillum. So here, Bacillus is rod shaped. Bacillus is rod shaped. Vibrio is comma shaped. Coccus is spherical. Coccus is spherical, and spirillum is spiral. Spirillum is spiral. So do remember this. This is very important. Then just give me a second. So these are the components of the cell envelope. These are the components of the cell envelope. We'll see one by one. Cell envelope is composed of three major parts: the glycocalyx, cell wall, and the cytoplasm. Glycocalyx, cell wall, and the cytoplasm. Remember this. Glycocalyx is a complex polysaccharide, which is the outermost layer. Its composition and thickness vary in different bacteria. It may be slime, that is loose, or it might be a capsule, which is thick and tough. Then the cell wall, it is the middle layer. It is very similar to that of prokaryotes. It gives the cell and gives the cell a proper shape and provides structure to the cell. It prevents the bacteria from bursting and collapsing. This is very important, bursting and collapsing. So plasma membrane, which is the inner layer, which is semi-permeable in nature and interacts with the outside. It is structurally similar to that of eukaryotes. Eukaryotes and prokaryote cell membrane are absolutely the same. Based on the types of cell envelopes and response to gram staining, bacteria are of two types: gram positive and gram negative. Gram positive will take up the stain and retain the stain, whereas gram negative, they do not retain the gram stain. They do not retain the gram stain. Then the next one, the infoldings of the plasma membrane in prokaryotic cell is called as a mesosome. It's called as a mesosome. The mesosome can be in the form of vesicle, tubules, or lamellae. Mesosome houses a lot of enzymes which are very similar to that of eukaryotes. So because they don't have any membrane-bound organelles, So usually what happens? The mesosome will perform many functions which has to be performed by other organelles. So remember that they help in cell wall formation, DNA replication, distribution of chromosomes to daughter cells, replication and secretion process, and increase the surface area of plasma membrane, and enzymatic content. Then they have chromatophores. Then to the nucleoid. Nucleoid here is synonymous to the nucleus. It is formed of non-membranous. That means there is no nuclear membrane and there is no nucleolus. And it has circular genetic material, a single chromosome genetic material, and a protein. Many bacteria have small circular DNA that are the plasmid outside the genomic DNA. It gives some unique phenotypic characters like antibiotic resistance to bacteria. So next, flagella. Flagella are thin filamentous extensions from the cell wall of motile bacteria. Their number and arrangement are varied in different bacteria. So the bacteria usually has three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside, and extends from the cell surface to the outside. Then pili and fimbriae. They are surface structures that have no role in motility. Pili, single pilus, are elongated tubular structures made of special protein pilin. The next one, fimbriae. Fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they help to attach the bacteria to rocks in streams and host tissues. Then ribosomes. Ribosomes are associated with the plasma membrane of prokaryotes. They are about 15 nanometer to 20 nanometer in size. They are made up of two subunits, 50 and 30s subunits. They together form 70s prokaryotic ribosome, whereas it is 80s in eukaryotes. Remember this. S stands for sedimentation coefficient, which is a measure for density and size. So S is a Swedberg unit actually. So ribosomes attach to a mRNA just after transcription, and they are called as polysomes, So do remember this. They're called as polysomes or polyribosomes. Then inclusion bodies. These are non-membranous structures which have stored the reserve food material seen freely in the cytoplasm of prokaryotic cells. Example, phosphate granules, cyanophysin granules, glycogen granules, and gas vacuoles. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. So now we'll look at eukaryotic cell. The main difference between eukaryotic and prokaryotic cell is the nucleus and membrane-bound organelles. So they have well-organized membrane-bound nucleus and other organelles. So the presence of membranes give clear compartmentalization of cytoplasm. They have complex locomotory and skeletal structures. Then cell organelles in eukaryotic cells. We'll go one by one. Cell wall. The cell wall is made up of, so we'll start with the cell membrane, Cell membrane, it's made up of phospholipid bilayer along with proteins and carbohydrates. Then they have a phospholipid with a phosphate head, which is a hydrophilic head, and a hydrophobic tail. The tails will face each other and the heads will project outwards. They have integral proteins and membrane proteins. Then the fluid mosaic model of the cell membrane was given by Singer and Nicholson in the year 1972. According to this, a quasi-fluid model of lipid enables the lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. So cell membrane has several functions. Transport of molecules. The membrane is selectively permeable to some molecules present on either side of it. Due to fluid nature, plasma membrane can help in cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Then there are different types of transport: passive transport and active transport. Passive transport, usually there is no expenditure of energy, so it moves from a region of higher concentration to a region of lower concentration, easy movement. Whereas active transport, there is expenditure of energy. Why? Because it has to be against the concentration gradient, This is very important. Then cell wall in plants is made up of cellulose and hemicellulose. Sometimes they have deposition of pectin and lignin also. Whereas cell wall in algae is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Okay. Now, a cell wall has initially has a primary cell wall, but as it grows, the primary cell wall also grows. But later, there is a secondary cell wall which is developed to the inner side of the membrane, Then the middle lamella is a layer mainly of calcium pectate. It glues different neighboring cells together. The cell wall and the middle lamella may be traversed by a plasmodesmata, which connect the cytoplasm of neighboring cells. Then functions. It gives shape to the cell. It protects the cell from mechanical damage and infection. It helps in cell-to-cell interaction. It acts as a barrier to undesirable macromolecules. Very important. Then the endomembrane system. The endomembrane system has different organs in it. Endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So in the endomembrane system, we have the endoplasmic reticulum, which divides the cytoplasm into two spaces. One is luminal space, and the other one is extraluminal space. Luminal space is inside the endoplasmic reticulum, and extraluminal space is the cytoplasm. So there are two types of endoplasmic reticulum. One is rough endoplasmic reticulum. The other one is smooth endoplasmic reticulum. Rough endoplasmic reticulum is involved with protein synthesis and secretion, whereas the smooth endoplasmic reticulum is involved with lipid formation, especially steroid hormones. So we'll go to the next one. That is the Golgi. Golgi was discovered by Camillo Golgi in the year 1898. In the year 1898. This is very important. The year 1898. It consists of flask disc-shaped sacs. It consists of flat disc-shaped sacs called as cisternae of 0.5 to 1 diameter. They are stacked parallel to each other. Cisternae are concentrically arranged with convex cis forming phase and concave trans maturing phase. Cis and trans phases are totally different but interconnected. So know this, very important. We'll go to the next one. It secretes materials to intracellular targets or outside the cell. Materials to be packed as vesicles from endoplasmic reticulum fuse with the cis phase and moves forward to the trans phase. This is why Golgi apparatus remains in close association with ER. Then the proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Then we go to the next one. Formation of glycoproteins and glycolipids. Then lysosomes. Lysosomes are also called as suicide bags of the cell. They are also called as suicide bags of the cell. So lysosomes contain almost all types of hydrolytic enzymes, that is hydrolases, lipases, proteases, carbohydrases. They are active as acidic pH. They are active at acidic pH. They digest carbohydrates, lipids, and nucleic acids and nucleic acid. So remember this. This is very important. Then we go to the next part. Just give me a second, students. So vacuoles are membrane-bound space found in the cytoplasm. A membrane-bound space found in the cytoplasm. So understand this. Then membrane found in the cytoplasm. The vacuoles are bound by a membrane called as the tonoplast. In plant cells, vacuoles can occur up to 90% of the volume of the cell. Then, so tonoplast will facilitate the transport of ions and other materials against the concentration gradient. That means whatever is less, it should be pushed to a region of higher concentration, that is against the concentration gradient. In amoeba, contractile vacuole helps in excretion. In many cells, example protists. Food vacuoles are formed by engulfing food material. Food vacuoles are formed by engulfing food material. Then the most important organelle, that is mitochondria. Mitochondria, mitochondria is also called as powerhouse of the cell. Is also called as powerhouse of the cell. Okay. One second. It's also called powerhouse of the cell because it produces ATP, which helps in production of ATP. Then mitochondria is a double membraned organelle. It has an outer membrane and a inner membrane. Outer membrane and the inner membrane. The space between the outer membrane and the inner membrane is called as intermembranal space. It's called as intermembranal space. To be very specific, it is called as perichondrial space. Perichondrial space. Then the number of infoldings of the inner membrane, they are called as cristae. Cristae. These cristae, what are there, will which are towards the matrix, and they increase the surface area. They increase the surface area. Then the two membranes have their own specific enzymes associated with mitochondrial function. The matrix possesses a circular DNA, okay, a few RNA molecules and ribosomes, that is 70s ribosomes. So always we study that eukaryotic ribosomes are 80s, but those present inside mitochondria and chloroplast, remember this, those present inside mitochondria and chloroplast, they are 70s, and components for protein synthesis. So they can prepare their own proteins. That is why these organelles are called as semi-autonomous organelles. Remember this, they are called as semi-autonomous organelles. The next one, plastids. They are found in all plant cells and in euglenoids. Remember this, they are found in all plant cells and euglenoids. They are large sized, easily observable under the microscope, and they contain some pigments. The next one, plastids. So plastids are of three types. So they are chloroplasts, chromoplasts, and leucoplasts. Chloroplasts have green color pigment chlorophyll. Chromoplasts help in bringing about color. Leucoplasts in storage of food. Then chloroplasts, they contain chlorophyll, remember this, and carotenoid pigments, and they trap light energy for photosynthesis. Okay. Then chromoplasts, they contain fat soluble carotenoid pigments like carotene, xanthophylls, etc. They give yellow, orange, and red color. Then leucoplasts, these are colorless plastids of varied shapes and sizes with stored nutrients. They include amyloplasts, which store starch, example potato, aleoplasts, which store oil and fats, and aleuroplasts, which store proteins. So remember this, very important for NEET. Then plastids. Chloroplasts are double membrane bound organelles. They have an outer membrane and an inner membrane. The space between the two membranes is called as intermembranal space. The space then to the interior to it is called as the stroma. In the stroma, you have many coin-like structures which are stacked one above the other, which are called as thylakoids. So the thylakoids will enclose a space called as the lumen. Then the chlorophyll pigments are present in the thylakoids. Remember this. Then plastids, in look at it, the characteristic feature is green color. So here the thylakoids are arranged in stacks called as grana or intergranal thylakoids. They are flat membranous tubules called stroma lamellae, which are connecting the thylakoids of the different grana. Understand this, it is also important. The stroma contains small double-stranded circular DNA molecules, ribosomes, and enzymes. Remember this, it has small double-stranded circular DNA molecule, ribosomes, and enzymes for the synthesis of carbohydrates and proteins. The ribosomes of the chloroplasts are smaller, that 70s, than the cytoplasmic ribosomes, which are of 80s. Understand this, which are of 80s. Then we'll go to the next part. Next one. Ribosomes. These are non-membranous granular structures composed of ribonucleic acid and proteins. So George Pallade was the person who discovered ribosomes in 1953. Remember this. This is very important. Okay, let's go to the next one. Just a second. So eukaryotic ribosome are of ATS type. They have two subunits, 60S and 40S. 60S and 40S. 60S has a larger subunit and 40S is the smaller subunit. So they together form ATS. Then the cytoskeletal elements. The cytoskeletal elements are made up of tubulin molecules which give stability to the cell. At the same time, when you look at the tubulin molecules, they are also involved in the formation of locomotory organelles, that is cilia and flagella. So remember that it is the same protein. So here we look at the locomotory organelle, that is cilia and flagella. Cilia are small structures which work like oars. So in the boat you would have seen, which will help in pushing the water backwards, oars, which help in the movement of the cell or the surrounding fluid Then flagella, they're longer, responsible for cell movement, and flagella of prokaryotes and eukaryotes are structurally different. So do you remember that basal body, hook and filament, or what I had done previously? So cilia and flagella are covered by a plasma membrane. They have a core called as the axoneme, has many microtubules running parallel to the long axis. Axoneme has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of central microtubules. This is called as 9+2 arrangement. So next we go to cilia and flagella. The central tubules are connected by bridges and are enclosed by a central sheath. It is connected to one of the tubules. Of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. The cilium and flagellum emerge from centriole-like structure called basal bodies. Then centrioles and centrosomes. So centrosome will contain 2 centrioles which are placed perpendicular to each other. Each centriole is made up of a protein, tubulin, which has 9 evenly spaced peripheral fibrils, very similar to flagella, but there are in doublets. Here we have triplets. Okay, so adjacent triplets are linked. Please do remember it has a central proteinaceous hub that gives a cartwheel-like arrangement. The next is the nucleus. So the nucleus was discovered by a scientist called Robert Brown in 1831. The materials of the nucleus, stained by basic dyes, was given by the name chromatin by Fleming. Normally, a cell has only one nucleus. Very few cells have multinucleated, and some of them don't have a nucleus. Example like RBC. Then the nucleus will contain nuclear envelope, nuclear matrix, chromatin, and nucleolus. Nuclear envelope is double layered with a space in between called as perinucleolar space. Okay, remember this, which is called as perinucleolar space. So inside to it is the nucleoplasm, in which we find the chromatin network and the nucleolus. The chromatin network will contain histone proteins and DNA, okay, which form the chromosomes later. Then nucleolus, one or more non-membranous spherical bodies are found, which is called as the nucleolus. It is continuous with the nucleoplasm. It is a site for ribosomal RNA synthesis. Then coming to the nucleus. In the nucleus, we have chromatin network, which will form chromosomes at the metaphase stage. The chromosomes have a constriction called as centromere. So So that is called as a primary constriction. So based on the location of the constriction, you have three types: metacentric, submetacentric, and acrocentric chromosomes and acrocentric chromosomes. So then we have the microbodies. These are membrane-bound minute vesicles that contains various enzymes present both in plant cell and animal cells. So please do remember this, students. Done. So, and also this one. Then so this part is very important, students. So have a look at this. So just give me a second. We'll start with MCQs. So let us all answer, students. So we'll go one by one. Cell theory was proposed by Schildin and Schwann. It was slightly modified by Rudolf Virchow. So it was by Schildin and Schwann. Then cell theory is not applicable for. Please answer. Cell theory is not applicable for virus. Virus is not considered a cell at all, so it can't be applied to it. Please answer this. Which of the following organism is not an example for eukaryotic organisms? So Escherichia coli here. Listen, students. Oh, 1 second. So Escherichia coli here is a prokaryote. It's a bacteria. Euglena is a eukaryote. Amoeba is a eukaryote. Paramecium is a eukaryote. Then, please answer this. Glycocalyx is associated with. Answer, students. Please answer. So glycocalyx is associated with. No, it is not associated with the nucleolus. It is not with the nucleus or the nucleosome. It is associated with the plasma membrane for the formation of cell envelope in prokaryotic cell. Answer this. Which of the following features is present in genetic system of prokaryotic cell? Please answer. Yes. Which of the following feature is present in the genetic system of prokaryotic cell? Understand this genetic system of prokaryotic cell. So here it is not mitotic division, it is not nucleolus, it is not histones, it is circular DNA. And also they have another circular DNA which is called as the plasmid. Very important one. Very good. Which of the following statement about prokaryotic cell is true? Please answer. Okay. See, which of the following about prokaryotic cell is true? It has membrane bound mitochondria. Wrong. So A is wrong. Mitochondria is not membrane bound. So they do not have a mitochondria. Okay. So how do we say that mitochondria is not membrane bound? So B is wrong. Okay. Then the nuclear material is not bound by a membrane. That is true. So they don't have a nuclear material. It does not have nuclear material by DNA and RNA. That is wrong. They either have DNA or RNA. So C is the correct answer. Good. Many students have answered it. Then seventh one. In eubacteria, a cellular component that resembles eukaryotic cell is. It should look exactly like the eukaryotic cell. Answer, students. Please answer. Okay. So we'll go one by one. Nucleus is wrong. Okay, it is not same. Why? Because they don't have nuclear membrane. Ribosome is wrong because 70s and 80s. Cell wall is also wrong because they have cellulose and hemicellulose. Here it's a different one. So plasma membrane is the correct answer. Both of them have the same type of plasma membrane. Then please answer this. Flagella of prokaryotic and eukaryotic cell differ in. Please answer. Yes, very good. So we'll go one by one. Type of movement and placement in the cell. Yeah, partially correct. Location in the cell and the mode of functioning wrong. Microtubular organization wrong. Microtubular it is. Yeah. Type of movement and placement in the cell. Type of movement and placement in the cell. In prokaryotic cell, the enzymes required for cellular respirations are located in. So they don't have any membrane bound organelles. Where are they located? The enzyme required for cellular respiration are located in. Please answer. So in prokaryotes, the enzyme required for cellular respiration are located in the cell membrane that is in the mesosome. That is in the mesosome. They don't have a mitochondria. The gas vacuoles are for storage. Plasmid is color. Then, which of the following statements regarding prokaryotic and eukaryotic cell is false? So answer, students. Which of the following statements regarding prokaryotic and Eukaryotic cell is wrong. Please answer, students. Please answer. So all prokaryotic cells do not have spindle apparatus. Look at this. So all prokaryotic cells do not have spindle apparatus. Yeah, that is true. So all of them don't have prokaryotic. They do not have nuclear membrane. That is also true. All eukaryotic cells have centriole near the nucleus. That is wrong. All eukaryotic cells have protective cell envelopes. So when you look at this, this will be slightly confusing. So all prokaryotic cells do not have spindle apparatus. That is absolutely true. Okay. So they've asked regarding prokaryotes and eukaryotes is false. All eukaryotic cells have centriole near the nucleus. Need not be wrong. All eukaryotic cells have protective cell envelope. Wrong. So when you look at this, nucleus, yes, I mean, in some at least it is correct. So we go with the D. All eukaryotic cells have protective cell envelopes. They don't have cell envelopes. Okay, cell envelope is found only in bacteria. Which of the following are inclusion bodies in prokaryotes? Please answer. Which of the following are inclusion bodies in prokaryotes? Which of the following are inclusion bodies in prokaryotes? So cyanophycean granules are inclusion bodies, phosphate granules, glycogen granules. So all of them are inclusion bodies in prokaryotes. Which of the following? Which of the following are absent in plant cells? So cell wall they have plastids, they have a central vacuole, they do not have a centrioles. They do not have a centriole. Centriole is not present. So they've asked which of them is absent in plant cells. So centrioles is the correct answer. Then in fluid mosaic model of plasma membrane, please answer. In fluid mosaic model of plasma membrane, so the upper layer is non-polar and hydrophilic. Hydrophilic is correct. Polar layer is hydrophobic. Wrong. Phospholipids form a biomolecular layer in the middle part. So A is the correct answer. Then structural lipids of cell membrane are, they are phospholipids. They are phospholipids. Then which of the following is not performed by plasma membrane? Please answer. Which of the following is not performed by plasma membrane? Plasma membrane, which one doesn't do it? Okay. Is not performed. So it anchors to the extracellular matrix which combine cells to form tissues. Yes, if it is animal cell, yes, it does. It contains the receptors that allow the exchange of information between cells. That is true. It aids the production of proteins for cellular activities. This is wrong. This is wrong. So it aids in transport of materials across the cell. Correct. So C is the correct answer. C is the correct answer. C is the correct answer. So we'll stop here, students. We'll take up a new chapter, that is structural organization in animals. Just give me a minute. Just give me a minute. Just give me a minute. oh sorry. So it is, there are three types of epithelium: squamous, cuboidal, and columnar epithelium. So squamous epithelium. So squamous epithelium is all isodiametric in shape. One second, students. I'll just show the picture. So they are very thin, flattened cells with irregular boundaries found in the wall of blood vessels, air sacs, and lungs. So they act as a diffusion boundary. Then columnar, then cuboidal epithelium. Just a second. Okay. They are composed of cube-like cells. So they are found in ducts of glands and tubular parts of nephron. Functions: secretion and absorption. The epithelium, the epithelium of PCT, okay, that is proximal convoluted tubule of nephron, will form microvilli. Then we have columnar epithelium, which are column-like cells, okay, which are found in the intestine. So they usually have microvilli, which will increase the surface area of absorption. So ciliated columnar epithelium. So they bear cilia. What are they done? They are present in the inner surface of hollow organs like bronchioles and fallopian tubes. Function: they to move particles or mucus in specific direction over the epithelium. This is very important. Then glandular epithelium. The cell is specialized for the secretion of two types: unicellular glands and multicellular glands. Okay, so consists of isolated glandular cells, that is goblet cells. Okay, example found in the alimentary canal. Whereas multicellular glands, they consist a cluster of cells, example salivary gland. Then based on whether they have ducts or not, they are classified into exocrine glands and endocrine glands. Exocrine glands are called as ductless glands. They do not have ducts. So example, they produce hormones. Then coming to compound epithelium, so they're composed of multi-layered cells. So they have a limited role in secretion and absorption. They cover the dry surface of the skin and the moist surface of buccal cavity, pharynx, inner lining of ducts of salivary glands, and pancreatic ducts. They function in the protection against chemical and mechanical stress. So between the epithelial cells, you'll have different types of junctions, that is the tight junction, adhering junction, and gap junctions. Tight junction, they help to stop substances from leaking across a tissue. Adhering junction, they perform cementing to keep the neighboring cells together. Then gap junction, they facilitate the cells to communicate with each other by connecting the cytoplasm of adjoining cells for rapid transfer of ions and molecules. Then we come to the connective tissue. Connective tissue, it links and supports other tissues and organs. So usually they have three components. One is the matrix, then you have the cells and the fibers. The first one, look at it, is loose connective tissue, areolar and adipose tissue. Dense connective tissue have dense regular and dense irregular. Then we have specialized connective tissue, cartilage, bone, and blood. So look at this loose connective tissue. It has cells, fibroblasts, macrophages, mast cells, etc. And the fibers are loosely arranged in a semi-fluid or ground substance. It includes areolar tissue, adipose tissue. Okay, areolar tissue, it has got macrophages, fibroblasts, and mast cells as cells. Then it has collagen fibers and elastin fibers, that is white fibers and yellow fibers. Okay, along with a background matrix. So adipose tissue is mainly concerned with storage of fat. The cells are called as adipocytes. Then in dense connective tissue, we have dense regular and dense irregular connective tissue. Dense regular connective tissue show regular pattern of fibers. Collagen fibers are present in rows between many parallel bundles of fibers, example tendons and ligaments. Tendons, they attach to the skeletal muscles of the bone. Ligaments, they attach to one bone to another. This is very important. Then dense irregular connective tissue are fibroblasts and fibers, mainly of collagen. This tissue is present in the skin. Then is cartilage. Cartilage is thin and pliable. Okay, pliable. Then the bones. Bones have a hard, non-pliable ground substance rich in calcium salts and collagen fibers, which give bone its strength. The bone cells are present in the spaces called lacunae. They are present in the spaces called lacunae. So what are the functions? It provides structural frame to the body, supports and protects softer tissues and organs. Remember this, this is very important. So the limb bones, example, leg bones, serve weight bearing functions. They take part in locomotion and movement, and blood cells are produced in the bone marrow, and blood cells are produced in the bone marrow. Then we come to blood. Blood is a fluid connective tissue containing plasma, red blood cells, white blood cells, and platelets. Okay. Then it helps in circulation of various materials. Then muscle tissue. Here, each muscle tissue is made up of many muscle fibers. Okay. Then they are composed of numerous fine myofibrils. The muscle fibers can contract, shorten, and relax. Okay. That is very important. Contract, shorten, and relax, lengthen. Muscles take part in locomotion and movements. Then muscles are of three types: skeletal, striped, or voluntary muscles, smooth, non-striated, or visceral muscles. Okay, then we have the cardiac muscle. So skeletal, striped, or voluntary muscles, they are attached to skeletal bones, that is biceps. Striations are present. So muscle fibers are bundled together and in parallel fashion. Remember this, they undergo fatigue. The next one, we go to a smooth muscle. Smooth muscles are small, spindle shaped, and they taper at both the ends. The cell junctions hold them together, and then they are bundled together in a connective tissue sheath. They are seen in wall of internal organs such as blood vessels, stomach, and intestine. Blood vessels, stomach, and intestine. Then cardiac muscle. They are involuntary muscles seen only the heart. Remember this, they are seen only the heart. Then they are involuntary, we cannot control them. They do not undergo fatigue, and in between the cells they have intercalated discs which allow them to work together. That is, all of the muscles in ventricles or in the auricles will contract together and relax together. Then coming to the neural tissue, this is for control and coordination. The individual units of neural tissue are called as neurons. Neurons are protected and supported by neuroglial cells. Understand this, neurons are protected and supported by neuroglial cells. Neuroglia makes up one half of the volume of the neural tissue, one half of the volume of the neural tissue. When the neuron is stimulated, an electrical disturbance, that is an impulse, is generated and will travel along the plasma membrane So we'll go to the next one. Organs and organ system. Tissues, many different types of tissues come together to form an organ, and many organs together form the organ system. So all of them will coordinatedly work in a individual. All of them will coordinated work in the individual. So now, morphology and anatomy. Morphology is the study of external features. Anatomy is the study of internal organs of animals. So, earthworm. It belongs to the phylum Annelida, class Oligochaeta, genus Ferritima, species Postuma. So usually the biological name of earthworm is asked, please do answer it. So here, let's look at the morphology. It is externally segmented. The first segment is the peristomium. The next segment is the prostomium. Okay, peristomium, sorry, it has a lobe called as the prostomium. The prostomium is sensory in function and is used to force open cracks in the soil into which the earthworm will crawl into. Then the mature earthworm has segments 14 to 16 covered in dark band of glandular tissue called as clitellum. The body has three parts: preclitellar, clitellar, and postclitellar segments. So there are three spermathecal apertures found in the 5th to 9th segments. Then there is a female genital pore found in the 14th segment. A pair of male genital pores are present in the ventrolateral sides of the 18th segment. Many minute nephridiopores open on the body surface. All the segments except the first and the last and the clitellum bear S-shaped setae, which helps in movement. Then the body wall. is out thin non-cellular cuticle. Then you have the epidermis, two muscle layers, circulars and longitudinal, then the internal organs. So digestion, you should remember here, you should remember the exact location of structures here. So the mouth, which is in the first segment, buccal cavity, first to third segment, muscular pharynx, fifth to seventh segment, muscular pharynx, fifth to seventh segment, stomach, 91 seconds student. So muscular pharynx, esophagus from 5th to 7th segment, muscular gizzard from 8th to 9th segment, stomach 19 to 14th segment, intestine 15th segment, intestine 15 to last segment, and then last is the anus. Last is the anus. Okay, so the gizzard helps to grind the soil particles. Then calciferous gland present in the stomach neutralize the humic acid in the humus. So we'll go to the next. So remember the intestinal CK and typhlosul. Typhlosul increases the surface area of absorption. Then circulatory system is closed. It consists of blood vessels. Remember this, capillaries and heart. So the contractions keep the blood circulating in one direction. Then there are blood glands on the 4th, 5th, and 6th segment, which produce phagocytic blood cells and hemoglobin, which is dissolved in the blood plasma. Then we go to the respiratory system. It is mostly by cuticle, that by the moist skin that is present. So excretory system is of nephridia. There are 3 types of nephridia: segmental nephridia, intertegumentary nephridia, and pharyngeal nephridia. So nervous system has a ventral nerve cord with ganglia in each segment, with a double ventral, with a ring which has a double supraesophageal ganglia, which are joined together to form the nerve ring. So then the sensory system includes light and touch sensitive receptors, and there are no eyes. Okay, there are no eyes. Chemoreceptors, taste receptors. Then a reproductive system. So they are hermaphrodites, that is bisexual. Males have testes, females have ovary. So the testes are lodged in the spermathecae, and comes out as the cocoon. Economic importance is earthworms are friends of farmers because they make burrows in soil and make it porous, which helps in respiration and penetration of the plant roots. This process of increasing soil fertility of the soil is called worm composting. It is also used as bait for fishing. Just a second, students. We'll go to the next part. So here, next one we have cockroach, that is Periplaneta americana. So cockroach body is divided into head, thorax, and abdomen. So the covered over by an exoskeleton, that is tergite and sternites. So remember this. Then the head has antennae, mouth parts, eye, which is a compound eye. It has biting and chewing mouth parts. Then the thorax has 3 parts: prothorax, mesothorax, and metathorax. Prothorax does not have wings. Mesothorax and metathorax have wings. So there are 2 types of wings. One is the forewing and the other one is the hindwing. The forewing are opaque, dark, and leathery, cover the hind limbs when at rest. The hind limbs are thin, membranous, and they are used in flight. So remember this. You can be able to differentiate male and female cockroach based on the anal style and also the size. Larger size, smaller size. Wings extend beyond the tip of the abdomen. They do not extend beyond the tip of the abdomen. Narrow abdomen, broader abdomen. Anal styles present, anal styles absent. This is very important. So just by looking at it, you can find out whether it is a male or a female. Then digestive system. So it has three parts: foregut, midgut, and hindgut. Foregut enters into the pharynx, then to the esophagus and the crop where the food is stored. Then at the end of the crop, you have a gizzard which has chitinous plates which will crush the food. Then you have a hepatic CK which will secrete enzymes into the midgut. Then it comes out to the Malpighian tubules. The Malpighian tubules help in excretion, then to the colon and to the rectum, and the opening is called as a cloaca. So here circulatory system is of open type, where blood vessels are poorly developed and they open into the hemocoel. Here the hemocoel will bath the visceral organs in blood, that is the hemolymph. Hemolymph is composed of plasma and hemocytes. Plasma and hemocytes. So remember this. So the respiratory system is of spiracles which extend into trachea into the animal. Then excretory system is through Malpighian tubules, and the type of excretory system is uricotelic because they excrete urea. The nervous system, they have a double ventral nerve cord with a brain at the end. Then cockroaches are dioecious, males and females are separate. The males bear testes and females bear ovaries. So after reproduction, they produce a cocoon which will lay eggs, which will have a lot of eggs. These eggs will open into nymphs, then form the adult. Then we come to the frog. Frog genus is Rana and species is Tigrina. It is a amphibian. So body is divided into head and trunk, forelimbs and hind limbs. Forelimbs have digits and hind limbs also have digits which are webbed. The digestive system is very similar to that of humans, okay, where digestion is by HCl and gastric juices, okay, and also pancreatic juice and bile. So even the intestine will bear villi, very similar to that of humans. Then respiration is by skin, buccal cavity, and by lungs, whereas in larvae it is by gills. Then circulatory system. The circulatory system has two parts, that is a blood vascular system and the lymphatic system. The blood vascular system, heart, blood vessels and blood. Lymphatic system, lymph channels and lymph nodes. Coming to anatomy, the circulatory system, there's a closed circulatory system. It has a 3-chambered heart, 2 atria and 1 ventricle. So here there is mixing of oxygenated and deoxygenated blood. So remember this, they have a hepatic portal system also. So excretory system, it has kidneys, ureters and urinary bladder. Frog is a ureotelic animal. That means it excretes urea. So nervous system has a central nervous system, peripheral nervous system and autonomic nervous system. So just remember it has cranial nerves and all that. And sense organs are touch, taste, smell, vision and hearing. So the reproductive system, males have testes, females have ovary. So the male's testis is usually attached to the kidney through fat bodies. Then they come out through the urinogenital tract and they open into the cloaca. Okay. Then females, the ovaries are near the kidneys. So there is no functional connection, whereas males have a connection through fat bodies. Here they don't know functional connection with the kidneys. A pair of oviduct from the ovaries open into the cloaca separately. A mature female lays around 2,500 to 3,000 ova at a time. Then fertilization is external and takes place in water. Fertilization is external and takes place in water. Then development involves a larva. Tadpole undergoes metamorphosis to form an adult. Remember this. This is very important. Next, economic importance. They eat insects and protect crop. The main ecological balance by serving as an important link in food chain and food web in the ecosystem. So in some countries they are used in as food. Okay. See, we have to be very fast. Now we'll solve some questions from structural organization in animals. Yeah, now I go. One second, Suraj. So we'll start answering. So the first question, the compound epithelium is found in the inner lining of. Compound epithelium is found in the inner lining of. Please answer, students. So they cover the dry surface of skin. Understand this, they cover the dry surface of skin. Okay. Then moist surfaces of buccal cavity, pharynx, inner lining of ducts of salivary glands and pancreatic ducts. So here it is the correct. Oviducts, wrong, because they have ciliated epithelium. Bronchioles also same. Proximal convoluted tubule has cuboidal epithelium. Cuboidal epithelium. Which type of epithelial tissue is found in endothelium of blood vessels? Please answer, students. Which type of epithelium is found in endothelium of blood vessels? Please answer. Which type of epithelium is found in endothelium of blood vessels? So the endothelium of blood vessel has squamous epithelium. It has a single layer of squamous epithelium. So remember this, squamous epithelium. Then internal walls of blood vessels and dash contain dash muscles. Internal wall of blood vessels and the dash contain dash muscles. Think and answer. So it is internal wall of stomach contains blood vessels. Why? Because stomach is a organ which has smooth muscles in it. So diaphragm is wrong. Diaphragm is wrong. Stomach and skeletal, no chance at all. So it is wrong. Next. Then in dense dash tissues, fibroblasts and collagen are arranged differently. These tissues are present in. Please answer. Please answer, students. So dense irregular tissues, fibroblasts and collagen are arranged differently. These tissues are present in skin. So B is the correct answer. Then blood is the medium through which oxygen is transported to different cells and tissues in the body. Which of the following process facilitates the exchange of oxygen between blood and cells in the body? So between blood and cells, what is the main method through which gases are exchanged? It is through diffusion. It is through diffusion. Then dash and adipose tissues are example of dash tissues. So areolar, so it is areolar. Areolar and adipose tissue are example of loose connective tissues. So A is the correct answer. Then seventh one. Dash muscle fibers are present in the internal lining of blood vessels. So it is smooth. So here B and D are gone, smooth. And it is fusiform shape and involuntary in function. So A is the correct, involuntary in function. This is important. Then eighth one. Eighth one. Answer, students. In dash, the collagen fibers are present in row between parallel bundles. It is an example of dense dash connective tissue. It has to be regular. So if they are arranged parallelly, it is regular. So this will go off. Okay. The information of which one will make the correct one. So it is tendon. It is not cartilage. It is not cartilage. It is tendon. Typhlosole is the internal median fold of the dorsal wall of earthworm's intestine. Intestinal typhlosole is present in between. Intestinal typhlosole is present in between 26th and 35th segment. It is between the 26th and 35th segment. Then 10th one. Dash nephridia are present on both sides of intersegmental septa. That is septal nephridia. Okay, that is septal. So pharyngeal is gone here. Septal nephridia. Okay, so of the dash segment, 15th to last. So here they have given third. So D is wrong. So A is correct. Okay, remember this. Very important. In mammals, the principal organ of respiratory system is the lungs. In cockroach, respiratory organ is, so skin doesn't help in that, gills doesn't help. Gills does help in few crustaceans, but very few. Lungs is wrong. So they have tracheae. So they have small openings called spiracles, which are closed with sphincters. So trachea is the correct answer. Then next one, the respiratory system is absent in certain organisms. These organisms respire through their skin by a process known as cutaneous respiration. Which of the following organisms breathes through cutaneous respiration? So through skin, whichever organism breathes, they've asked. So fish is wrong because it breathes through gill. Cockroach is wrong because it is through spiracles. Grasshopper is also wrong, same as then. The correct answer is earthworm. Then 13th one. The function of spermathecae in earthworm is, so spermathecae will actually store sperms. It will store sperms. That is very important. So storage of own sperms during copulation, it is wrong. Storage of own ova and sperms, wrong. Storage of sperms of other earthworm during copulation, that is correct. Storage of embryonic nutrients, wrong. So if the cocoon slips over the posterior part of the earthworm, so what will happen? So if it is only going through the anterior part, then only it will get fertilized. If it comes to the posterior part, it will not get fertilized. Okay. So zygotes will be destroyed. Zygotes are obviously not formed because there is no fertilization. Fertilization will be normal. This is also wrong. Egg will not be fertilized. This is true. Both the ends will remain open. Wrong. So eggs will not be fertilized. This is true. 14th one, C is the correct answer. Then 15th one. More developed endoskeleton in cockroach is found in. So abdomen is wrong, legs is wrong. It is actually the head. Because they have lot of parts moving around in the head. Then wings of cockroach are attached to. So mesothorax is wrong because they have two pairs, two segments have to be there. Pro is wrong. Mesothorax and metathorax is the correct answer. Then the image formed in compound eye of cockroaches. So compound eye of cockroach has many individual units called as ommatidia. Okay. So remember this. It is a mosaic image. It is not a straight image. It is not a reverse image. It is not a image to be formed at all. It is a mosaic image. Then the number of thoracic and abdominal ganglia are. So the number of thoracic and abdominal ganglia are, think and answer. So how many ganglia can be present in the nervous system? So obviously thoracic would be three ganglia. So this would be wrong. So think and answer which would be correct. It is actually D. It is actually D. It is not 10. They don't have 10 ganglia. They have only 6 ganglia in the abdomen. Then development of frog is, see, if it is direct development, the young ones will resemble the adults. If it is indirect development, the young ones will not resemble the adults. Can you please answer? It is indirect development because they have a larval stage, which is called as a tadpole, larva. Then opening of rectum in frog is called, the opening of rectum in frog is called, answer students, the opening of rectum in frog is called cloaca. So wrong, coccyx is wrong, none of the above. So cloaca is an opening for GI tract, that is gastrointestinal tract, urinary tract, and genital tracts. So urinogenital and gastrointestinal, all opening, there is a single one, which is called as cloaca. The lining of intestine and kidneys in humans is, the lining of kidneys, intestine and kidneys is, please answer. It is not keratinized. Keratinized means on the normal skin, what we have is keratinized epithelium. Brush border is wrong. So they have asked lining of intestine and kidneys. So they have normal cells. So brush border, they have microvilli. Ciliated is wrong. None of the above is correct. So some students will say, sir, it is brush border because nephron, they'll take into PCT into consideration, but that is wrong. They've taken the whole of kidney. The type of cell junction which facilitates cell to cell communication is, please answer. It is gap junctions. Gap junctions help in cell to cell communication. Tight junctions, so it prevents from leaking. So no leaking here. Okay. Adhering junctions. So adhering junctions, the main function is cementing. Okay. So remember this. This is very important. Then we go to the next one. Keratinized dead layer of skin is made up of. So keratinized dead layer of skin is made up of. It is made up of stratified squamous epithelium. So squamous epithelium which is stacked one over the other. So the skin that is present on us is keratinized squamous epithelium. Then histamine, serotonin, and heparin are secreted by. Histamine, serotonin, and heparin are secreted by. They are secreted by basophils. So eosinophils, they are in allergic reactions, but secreted by basophils, which are in inflammatory reactions. Then, which of the following cells is not phagocytic? Answer. See, macrophage and monocyte, they're almost the same. Monocyte will only transform into a macrophage, then neutrophils. So it is basophil, which is the correct answer. Then, which of the following human cells do not contain mitochondria? Which of them does not contain mitochondria? So answer students, it is the red blood cells which do not contain mitochondria. The red blood cells do not have nucleus, they do not have endoplasmic reticulum, they do not have Golgi, and they do not have mitochondria. That is why they're usually called as corpuscles. 27. Which of the following prevents the conversion of prothrombin to thrombin in undamaged blood vessel? So what should it be? It should be an anticoagulant. It should be an anticoagulant. So what is the anticoagulant here? It is heparin. It is heparin. Then 28th, clitellum is found in. Where is the clitellum found in earthworms? It is in the 14th, 15th, and the 16th segment. And the segments in front of it is preclitellar, and the segments after it is postclitellar segments. Then, which of the following is correct pairing of body part and the kind of muscle that moves it? So answer students, heart wall is involuntary, correct, but it is striated, so it can't be right. So biceps of upper arm, smooth muscles, wrong. Abdominal wall, smooth muscle, this is correct. Iris, involuntary smooth muscle, wrong. So iris is not by smooth muscle, that is because of ciliary elements. So C is the correct answer. C is the correct answer. So we'll stop here, students. So Rajul sir. I think it's time up and we'll stop here, students. Thank you.